Hello everyone. This is lecture 20, part 1. In this lecture we're going to be taking a look at the seed plants. Traditionally, seed plants are recognized as a subgroup of vascular plants. All seed plants are vascular plants. So the vascular plants, formerly known as tracheophytes because they contain true vascular tissue, i.e. xylem and phloem, are divided into two major groups: the seedless vascular plants, and these are represented by the ferns and the fern allies, such as horsetails and lycophytes. And the other major group of vascular plants are the seed plants, also known as spermatophytes, because these plants reproduce via seeds. Among the seed plants, there are two major groups: the gymnosperms and the angiosperms. There are currently 4 phyla of gymnosperms that are recognized: the conifers, the cycads, the ginkgos, and the gnetophytes. And then the other major phylum of seed plants are the angiosperms, or the flowering plants. What do we mean by gymnosperm and angiosperm? What do these terms refer to? Essentially, gymnosperms and angiosperms differ in the way that the seeds are displayed or how they occur on the plant. If we look at the terms gymnosperm and angiosperm, we can understand this a little bit better. Gymnosperm is a term that means naked seed or exposed seed, and this refers to the fact that the seeds on gymnosperms, if we consider, for instance, say a pine tree, the seeds on a pine cone sit on top of modified leaf structures known as scales. The scale is part of the cone or the strobolus, and the point is that the seed does not have any additional tissue derived from a flower or anything like that that makes up the entire unit that carries the seed. So it's an exposed seed. You can see something similar in this image on the right. This is a cone or strobolus of a plant known as a cycad, a very primitive type of gymnosperms. And the little green structures that you see are the seeds, and they literally, when the cone is mature, the cone breaks open and the seeds basically just spill out of the cone. There's no additional tissue that surrounds those seeds. So gymnosperm, naked seed. In contrast, angiosperm, this term is translated as enclosed seed or seeds in vessels. And what those vessels are, they're little chambers, you can think of them, for the seeds that are derived from a part of the flower known as the ovary. So when we study flowers, you'll see the ovary is where the seeds are housed, and later the ovary will develop into a fruit once the seeds are mature. And you can see that in this image of the apple. Apples are flowering plants or angiosperms, and the seeds are enclosed in this structure in the middle. That's the ovary. In fact, in apples and other members of this family, the rose family, there's even additional tissue derived from the flower known as the hypanthium. That's not present in all flowers and fruits, but it just happens to be the case with this example. And in order to set the context for this question, we have to think about the more primitive plants that we've already studied. So bryophytes and seedless vascular plants, they reproduce using a process that I would refer to as the spore habit. In other words, they release free-living spores, usually from a structure like a sporangium, or in some of the more advanced plants like, say for instance, lycophytes and horsetails, a structure called a strobolus. But the point is that the sporangia on these plants opens up and releases spores. Those spores are single haploid cells. They don't have any reserve tissue or nutritive tissue, And basically, in the picture here, these represent spores from a fern plant. And so these spores have a short period of time before they have to germinate and begin doing photosynthesis right away and develop into a free-living gametophyte, which eventually will produce gametes. So that's the spore habit. The seed habit is very different because, well, seeds are a lot more complex than spores. Basically, a seed contains a multicellular diploid embryo that has already reached a stage of development where all of the major tissues and organs that will be present in the plant have been formed. And in addition to that, that embryo has a reserve tissue or a nutritive tissue. The nature of this nutritive tissue will vary whether we're talking about gymnosperms or angiosperms, but essentially this means that the embryo has a food supply for growth and it doesn't have to start doing photosynthesis right away. Another important aspect of seeds is that they enter a condition known as dormancy. And so unlike spores that only have a short window of time before they have to germinate and grow, seeds can remain dormant in many cases for years, decades, even centuries. There have been seeds that have been isolated from archaeological sites that are thousands of years old that the seeds are still viable. So it's a really remarkable feature of seeds that even though you know, they're in a dormant state, a suspended state, they're still viable, they're still alive, and they have the potential oftentimes to germinate and grow into a new plant. So what we want to look at then, since the seedless plants were the first plants or the earliest plants, there's no question that seeds had to have evolved from some group of seedless plants. And in fact, botanists believe that this process probably occurred multiple times. And so there is probably no single origin for seed plants, which makes the classification of the seed plants a little bit challenging because, again, they represent probably different evolutionary lineages. But in all cases, certain things had to happen for seeds to evolve. Now you can understand that reproduction by seeds would have a lot of advantages over reproduction by spores. We just talked about the fact that seeds can remain dormant, that they have a storage tissue that can feed the embryo, that the embryo is basically like a miniature version of the adult plant. But another important aspect of seeds is that because of how sexual reproduction works in seeds, seed plants do not depend directly on water for fertilization. And what we'll see is that the female gametophyte, the egg-producing stage of the plant, will remain attached to the parent plant. And instead of sperm having to swim to the egg through water, which is what you find in bryophytes and seedless vascular plants, instead the sperm are basically delivered to the egg via pollen. And that is what pollen is. It's a sperm delivery system. And there's different mechanisms by which pollen can be delivered, so-called pollination. Again, we'll talk about some of these mechanisms in a subsequent lecture. In most gymnosperms, the plants are pollinated by wind. So the plants make a lot of pollen and it's dispersed by wind, and some of that pollen lands on the female plant or the female part of the plant and carries out reproduction. In many flowering plants or angiosperms, animals act as pollination vectors, and these are some of the well-known insect pollination mechanisms primarily that is one of the really fascinating aspects of flowering plants. So in any case, the bottom line is that in this type of reproduction, fertilization no longer requires external water. You can think of it, this is like an internal form of fertilization. So that obviously allowed these plants to inhabit drier environments and colonize a much larger area than the seedless plants, which are usually quite restricted to fairly wet climates. Now, as I mentioned, there is not a universal consensus on how the seed plants evolved. The fossil record is a little bit confusing on this. Seed plants had appeared on Earth by about 350 million years ago. So this is a relatively short period of time after the first land plants. Recall that the first land plants appeared about 420 million years ago. And so from these earliest plants, seed plants evolved relatively quickly. And that would point to, again, the advantages of reproduction using seeds instead of spores. But we don't really know which group of seedless plants gave rise to modern seed plants. And in fact, in the fossil record, there's evidence for several groups of plants that were probably early seed plants, but that have since gone extinct. And so which one of these extinct groups of plants, or maybe several extinct groups of plants, gave rise to modern seed plants, that's a question that botanists have not been able to answer yet. There were two kind of large groups of plants that were present, again, some 350 to 400 million years ago, that represent possible ancestors to modern seed plants, although again the evidence is a little bit sketchy. One group is called Gymnosperms. These are plants that looked a lot like modern gymnosperms. They were tall trees, woody plants, had a vascular cambium, but closer inspection of some of these extinct Gymnosperms, and a classic example is a plant known as Archaeopteris, reveals that they don't actually have seeds. They have sporangia, a bit like the sori of ferns that occur usually along the leaves, on the outside of the leaves. So the Gymnosperms, although they were tree-like and had some superficial similarities to modern gymnosperms, it's thought that they probably were not the ancestors of the seed plants. So there's another group of plants, and these are known as seed ferns. And as the name suggests, these morphologically look more like ferns. They had fronds. Many of them did not have secondary growth. But a closer look at how they reproduced reveals that they actually had primitive versions of seeds. And essentially, the key difference is that there were sporangia on these leaves, and the sporangium, along with the spores and the gametophyte, all remain on that parent plant and develop into a seed, rather than releasing spores and having a free-living gametophyte stage. Now, again, in modern systematics, the seed plants are probably, again, not a monophyletic group. It means that they're either paraphyletic, meaning that again, we have an ancestor, but certain descendants are excluded, or possibly even polyphyletic, where we have the seed habit appearing multiple times in evolution. So again, we don't know the answer to that question at this point. But if we are to try to understand how seeds evolved in the first place, several things had to occur to make the transition from a spore habit to a so-called seed habit. And those are listed on the slide here, and I'll go through each one of these in a little bit more detail. Number one, you had to have heterospore. Heterospore, two types of spores, two types of gametophytes. Number two, you had to have something called endospore, where the gametophyte develops within the spore case. Number three, on the female side, you had to have an attached gametophyte. So the gametophyte remains attached to the parent plant. It does not have a free-living stage like you see in ferns or bryophytes. And that gametophyte has got some additional layers of cells around it that are derived from the sporophyte. These are the seed coat, which are known as integument's, and the nucellus, which is essentially the wall of the sporangium. Furthermore, the seed, in order to, or for the embryo to grow, at least to a stage of development where the seed would be viable, that embryo had to have some type of nutritive tissue or reserve tissue. So this was provide food for the embryo as it develops. And then finally, you had to have a mechanism to get sperm to the egg, and that's what I referred to in an earlier slide, essentially is what pollen is. Pollen is a means to carry out fertilization without the need for external water. So let's review each one of these and make sure we understand what each of these terms means. So heterospermy, as you recall, is a condition where you have two types of sporangia, a male sporangium known as a microsporangium and a female sporangium known as a megasporangium. The microsporangium makes microspores through meiosis. The megasporangium makes megaspores also through meiosis. And the microspores and the megaspores then develop into microgametophytes or male gametophytes, which is basically the pollen grain, and megagametophytes, which will eventually become a structure known as the ovule, and that's what develops into the seed. So the microgametophyte produces sperm. In seed plants, the sperm are produced, there's no antheridia in the sperm, in the microgametophyte. Instead, it's a highly reduced form of the gametophyte stage, and it's just a few cells, and one or usually sometimes two cells serve as so-called germ cells that will produce the sperm. And then the megagametophyte produces eggs. In gymnosperms, the megagametophyte retains the archegonium, but as we will see in the flowering plants, that structure, the archegonium, is lost. And so again, you have a much more highly reduced gametophyte stage. So heterospermy, as we have already seen, actually occurred in the seedless plants. There are some seedless vascular plants, in particular in the lycophytes. Recall there's a plant that was reviewed known as Selaginella, and that's a classic example of a lycophyte, again a seedless plant, that makes these two separate types of sporangia and gametophytes. It's shown in the image here. The structure that you see on the left, this is a Selaginella, part of a Selaginella strobilus, and the image, the left part of this image shows the megaspores forming within the megasporangium, and on the right we have the microspores forming within the microsporangium. And something very similar to that was passed on to the early gymnosperms. Endospermy is the development of the gametophyte within the spore case. So unlike the seedless plants where the spore is released from the sporangium, and then that spore germinates and the gametophyte develops outside of the spore case, that's called exospermy. In this case, endospermy, the gametophyte develops within the spore case. And so what you see here is an example of a spore. Again, this is from Selaginella, which is a seedless plant, highlighting the fact that some of these steps occurred in the seedless plants. This outer layer that you see is the spore wall, the megaspore wall, and then the gametophyte has developed inside of that from a megaspore. It consists of a cellular region shown at the top, and the cellular region is where you would have an archegonium and an egg, sometimes more than one, and then the lower part is so-called undifferentiated megaspore cytoplasm that again will play a role in seed development. Similar situation on the right. This is a microspore. The microspores, as the name suggests, are much smaller than the megaspores. And inside the microspore, you have microgametophyte development, and really it's just a few cells. Some of these cells are called prothallal or vegetative cells. Again, it varies depending on the group of seed plants that we're talking about. And we'll look in more detail at the life cycles of certain gymnosperms like the conifers. But essentially you have a vegetative cell and then a generative cell that produces the sperm. And so there might be a primitive antheridium, but again, once we get to the full-blown seed plants, the antheridium has been lost. There are in Selaginella and in certain seed plants, certain primitive seed plants, the sperm have retained flagella, interestingly. But as plants became more advanced, that trait was lost, and so the sperm do not have flagella. Instead, the sperm are delivered to the egg through a structure called the pollen tube, and we'll look at the pollen tube in a few minutes. So the third aspect of seed evolution involved the megagametophyte remaining attached to the parent plant. And you can see that in this image here. This is actually an image of a pine scale. So imagine a female pine cone, and the scale is where you find the seeds on the surface of those scales. And so this structure that you're looking at, that I'm highlighting, is the growing seed. Okay. At this stage, it's not called a seed, it's called an ovule. So an ovule, you can think about that as an immature seed. And what do we find in the ovule? Well, the outer part of the ovule is a structure known as integuments. Integuments will become the seed coat, and that is derived from maternal tissue, from the sporophyte of the mother plant. Just inside that, you have a layer of cells, or a tissue layer known as the nucellus. That's the wall of the megasporangium. And remember that gametophyte development occurs inside the spore case. So you have a single megaspore that develops into a gametophyte within the nucellus. And that's what we see in the center of this structure. This is the megagametophyte. So you can see it's filling up the nucellus, the cavity, and these two eye-looking things that you see near the top, those are the archegonia, and they will produce the eggs. So this entire structure remains attached to the parent plant. It receives nutrients from the parent plant, and there's never a free-living megagametophyte stage in the seed plants. So another aspect of this is a storage tissue or a nutritive tissue to feed the developing embryo. Once again, the embryo, it's not going to be able to produce its own food through photosynthesis because at this stage of development there is no photosynthesis going on, and so instead it has to obtain nutrients from another source. And that other source, again, we'll talk about that as being nutritive tissue. It is going to provide carbon and energy and nutrients for the developing embryo. And in the case of gymnosperms, the nutritive tissue is actually the megagametophyte itself. So once fertilization happens in the archegonia, once the eggs become fertilized, that embryo begins to develop, and it's receiving its nutrients from the remains of the megagametophyte. All right, so just remember that the megagametophyte serves as the nutritive tissue in the gymnosperms. What we're going to find is that in the angiosperms, they adopted a very different mechanism for producing nutritive tissue, and this mechanism is thought to have been one factor in accelerating the life cycle that we find in most angiosperms that have a much shorter life cycle than gymnosperms. And then finally, the last aspect of seed evolution, as I mentioned earlier, is a mechanism to carry out sexual reproduction. And so imagine you have a tall tree and the ovules are developing in cones at the top of the tree. It would be impossible, presumably, for sperm to swim from one tree to another in that type of growth habit. And so fortunately that is not what needs to happen. Instead, the sperm are delivered directly to the egg via pollen. And so the pollen is what's forming in the microsporangia. Okay, so again, if you imagine a pine tree, the little yellow cone-looking things that come out in the spring, those are the male cones. They make the pollen. So the pollen is the microgametophyte. And in most gymnosperms, the pollen is very light and dry, and so it's easy for wind pollination mechanisms. And so that's why we have clouds and clouds of pollen from pine and other wind pollinated trees in the spring. Some of that pollen will reach the ovules on the female side, sometimes on the same tree, oftentimes on different trees, and that is pollination. So the delivery of pollen to the ovule, and once that happens, then sexual reproduction can occur in the ovule, and that's how you get a seed. So let's just take a quick look at that. This slide is showing the development of the ovule at different stages. And so again, the ovule is the megagametophyte and the surrounding tissues from the mother plant, especially the integuments, okay, that outer layer. And at this stage of development, this is a very early stage in development where the megaspore has just formed. Usually in the megasporangium, there's just one megaspore that forms from meiosis. And so now the megaspore has formed, and it will begin to grow inside the ovule and develop into the female gametophyte or the megagametophyte, shown in this kind of light yellow tissue in the center. Once that female gametophyte has reached a certain stage, an archegonium will form, and inside the archegonium you'll have eggs. And again, it can vary, but usually one or sometimes several eggs will form. So those are the female gametes. And recall that what needs to happen is, in order for that egg to develop into a new plant, all right, through sexual reproduction, pollen has to be delivered, and that occurs from the pollen grain. And some pollen grains will, there's an entrance to the inner part of the ovule, and this entrance is known as the micropyle. So the micropyle is a little gap in the integuments, and a pollen grain can pass through that gap, and once the pollen grain has landed at the base of the ovule, it will form a pollen tube, and the pollen tube is the actual delivery mechanism for the sperm. The sperm cells travel down that tube, and eventually one of them will fertilize the egg. So it's actually the pollen tube specifically that eliminated the need for external water for sexual reproduction to take place in the seed plants. After fertilization occurs, and again, this is a very slow process in gymnosperms. It can take several years for a pine tree, for instance, to go from an immature female cone to a fully developed seed. But eventually that fertilized egg will develop into an embryo. That's the new sporophyte, and the seed will enter a stage of dormancy. The embryo will be, as it's growing, will be fed by the female gametophyte or the megagametophyte, which serves as the nutritive tissue. So all of those things together are the steps that were necessary for seeds to evolve from plants that use the so-called spore habit for reproduction. And just to kind of show you a few images to highlight this process. And this picture, this is a close-up picture of a female ovule in pine. Okay, so this is basically the two sort of outer layers of cells. Those represent the integuments. This little opening that you see down on the bottom right is the micropyle. So that's that little opening in the integuments. You can see a few pollen grains have actually crossed through the micropyle, and now they've landed at the base of the ovule. And so this tissue that we see right here is megagametophyte tissue, and you can see down embedded in this tissue is the archegonium. Again, it's a very simple structure in seed plants, if they have them at all. And in the archegonium, you'll have the female gamete or the egg. And so once these pollen grains land at the base of the ovule, one or more of them will form a pollen tube, and that pollen tube has to essentially burrow down into the megagametophyte, and eventually it will reach the archegonium and the egg, release the sperm, and fertilization will occur. And once fertilization occurs, then the zygote will undergo development into an embryo, and after that process goes on for, in some cases, many months. In the case of pine, it can even an entire year, but the seed will eventually become mature, and at that point, seed development has stopped. And so in these images on the upper left, these are pine seeds, and I'm sure we've all seen these. So that the little oval-looking structures of the seeds, and in pine they have a little wing that attaches to the seed that helps the seeds disperse when the cone breaks open. On the bottom you see a young germinated pine seedling. So this is a seed that may have been in the soil in a dormant state for many years. Again, it depends on the species, but pine seeds can retain a viable seed bank in the soil, usually for many years. And then, when conditions are right in terms of temperature and moisture, the seed can germinate. You can see the young pine tree developing. It's interesting to consider how many generations are represented in one of these pine seeds. So if we start with the outer part of the seed, which is the integument, or the seed coat develops from the integument. That is sporophyte tissue from the mother plant. You can think about that as grandmother tissue. And then once you open up the seed, the bulk of the seed is actually the megagametophyte. So the megagametophyte has formed as a result of meiosis and then development of the spore, the megaspore. And so this is the haploid stage after, the sporophyte. So that would represent the maternal or the mother generation. And then in the center here you have the young embryo. Again, you can see it looks a lot like the plant that will eventually germinate. So you have the root shoot axis with the hypocotyl and numerous cotyledons. That represents the diploid embryo that form from fertilization. So there's actually 3 generations that are represented in this in the seed of a gymnosperm. So finally, just to wrap up this lecture on the evolution of seeds, we can understand that the seed habit would have conferred a number of advantages over the spore habit and allowed plants that evolve seeds to become much more successful ecologically compared to their seedless counterparts. And so some of those advantages are listed here. You have advantages before fertilization, so essentially, again, before the seed begins to form, since the megagametophyte is attached to the parent sporophyte, it basically has a protected stage in its life. It's protected from harsh environments. It receives resources and nourishment directly from the sporophyte, and finally, when sexual reproduction is ready to occur, water is not needed because pollen will be delivering the sperm to the egg, so water is no longer required for fertilization, again, unlike seedless plants. After fertilization, many of these same advantages still apply, so now it's the embryo that is attached to the sporophyte, and as before, the embryo is protected from harsh environments. There's a connection between the sporophyte and the gametophyte, so although the embryo is receiving food directly from the remains of the megagametophyte, there's still a transfer of resources from the sporophyte, so that is again going to help with early development of the plant. And then of course another big advantage is that once development has reached a certain stage, the seed enters a physiological state known as dormancy. And dormancy can last for a very long time. And so this allows those seeds that have formed through sexual reproduction to remain viable in the environment, and germination can be delayed until conditions are ideal for the plant to grow. And this is why, again, the seed plants so much more diverse, generally speaking, than the seedless plants.